Well, hello everyone. My name is Helena Bowman, the new president for this chapter, California Native Plant Society. And for you who don't know anything about the chapter or the organization, we have about 35 chapters all over California. It's a volunteer organization, and we are all interested in conserving the plants, planting native plants in our yard, or learn how to identify and learn to the plants. I study botany in Sweden, which has been covered by the last ice age, and we only have 300 and I wrote it down here, pardon me, 372 plants. So I know them all by heart. Then I moved to California, and here we have 6,500 plants. So I don't know all the plants. I'm working on it, but I'm just happy when I go out and hike and see a beautiful flower. So I want to acknowledge too that we are residing on the ancestral lands of the Chumash people and the Tonga land. Let's see what I have here. I want to thank our board members, especially the ones retiring from the board. That is Cliff McLean, the last past president, and Jane Tyrrell as a treasurer, and Barbara Eisenstein, which was the plant sale wizard.
Oh no, Leah's here.
So the new board that I have is, I'm president, vice president is Orchid Black, secretary is On Wang, treasury, the new treasury is Joy Waters, and our chapter delegate is Margaret Gallagher. We also have a social media chair, William Hallström. He will take care of the Facebook and the Instagram and reworking that. So with this board being changed, this means we need a lot of new souls to help out to fill the positions we need. Especially, I'm interested in starting field trips again to replace, since we can't meet in person. I think field trips will help us connect more to us in person. And we have other important places that need help. Plant sale. We have thoughts and started out planning to have the plant sale again in Monrovia. So anyone who's interested to work with the plant sale, you have to contact us and we'll get a committee started real soon. So keep a close look at our website. I will put out information there and what we need. And I think I also have here, you have to be patient with it. Take a while to get used to being in a chair here again. Also want to thank Rebecca Shield Moose for being our tech host tonight. And I think I just turn it over to Orchid Black, who will introduce our speaker, Mr. Stu Pickell.
Thanks, Helena. I'd like to let everyone know that to please put your questions in the chat, but Rebecca will collect all the questions and give them to Stu at the end. So he may take questions in the middle, but if your question does not get answered, it should be answered at the end with all the rest of the question. So Stu Pickell is a slime mold and fungi enthusiast currently residing in Pasadena. His interest in fungi began while he was attending college in Portland, Oregon, after becoming aware of the, I love this phrase, enormous fungal biodiversity of the Pacific Northwest. So I guess that's fungi everywhere. Since moving down here, he's been documenting and photographing with amazing photographs. You will love them. Often overlooked species because they're tiny of fungi and myxomycetes, and focusing on riparian areas of the San Gabriel Mountains. You can find Stu's slime mold and fungi photos on Instagram at Stu Cucumber bottom hyphen, or space. So And someone will put that in the chat for you. Thank you. And without further ado, I'm going to hand it over to you, Stu. And you are muted.
I'm here. Thank you, Marky, and thank you all for having me here tonight to talk to you about something that really has nothing to do with native plants much at all. But it's something that I find very fascinating, beautiful, and I'm excited to share. So, as Orchid mentioned, my name's Stu Pickell. I consider myself a fungi and slime mold enthusiast. I don't study these organisms in any formal capacity. I love documenting and photographing them. I first got interested in them on a trip to Washington State about 4 years ago when I found one while looking for mushrooms. I've been hunting mushrooms for about 11 years. And I was kind of just captivated by its intricate beauty and its colors. I realized when I came back to Los Angeles that, well, I had known for a while that mushrooms weren't as easy to find as they were in the Pacific Northwest. When I lived in Portland, it's much drier here. Even on a good year, there's not a whole lot of mushrooms. But slime molds you can kind of find all year round. If you know where to look, they're quite common, just overlooked. And it became kind of a stand-in for my obsession with fungi. Most of the photos I'll share tonight, or all of them, with the exception of the ones I know otherwise are my own. Most of them, nearly all of them, are from the San Gabriel Mountains and the surrounding area. So I just want to share with you first a little bit about slime molds or myxomycetes and what they are, what they're all about, and then share with you some of my favorite finds and a little bit about how to find them here and what to look for. So what are slime molds? The myxomycetes or plasmodial slime molds are a class of amoeboid protists, which means they're single-celled eukaryotic organisms, meaning they're not plants, animal, or fungi, but their cells have a nucleus, and they can change their shape. So single cells originally with single nuclei fuse into macroscopically visible multinucleate cells that have many, many nuclei, such as this one on the right. This is actually a single cell called a plasmodium, or plasmodia plural. These plasmodia kind of crawl around leaf litter, the forest floor, logs, all kinds of different plant matter substrates, looking for food. Now, they have characteristics of both animals and fungi. They're like animals in the sense that they're locomotive. They creep around looking for food. This movement is something we usually only associate with animals. And they're like fungi in the sense that they produce spore-bearing structures in their reproductive phase. They spend most of their lives in this plasmodium stage that you see here on the right. You can see why they get the name slime mold. They're kind of slimy, viscous. They engulf bacteria and other bits of organic matter and digest it. Most of the ones I'll show tonight are quite small. Here on the left, we have a photo that I took with my phone of a species, Trichidicipiens, with a human head for scale. And on the right is a close-up of that same, one of those same sporangia, or two of them. Sporangia is another name for fruit bodies. That I took with my camera. So you see they form these stalks, some of them, with balls that contain the spores. And it's in some ways similar to a mushroom. Often it's mistaken for one. And you'll see in the mushroom ID groups on Facebook, people will first find out about slime molds by misidentifying one as a mushroom, asking for an ID only to find out that this is not a fungus at all. It's amoeba. So most of the photos I'll show tonight are of fruit bodies that are 1 to 2 millimeters tall, which is about the average size for most of these, the fruit bodies, the spore-bearing structures. Here is a diagram from a textbook on slime molds. It shows where they fall into the eukaryotic tree of life. You can see them under amoebozoa with that red arrow pointing to them. And hopefully, unsurprisingly, based on what I've said so far, they fall somewhat closely to both fungi and animals. As slime molds are classified right now, they fall into 5 orders, like Cytosteales, Lyciales, Physareales, Trichiales, and Stemonitales, which contain 11 families, 52 genera, and about 1,000 known species worldwide. So when I decided to give this talk, I wasn't sure what to say to a group of plant people. I've been asking myself this question: how do I make slime molds relevant to people who are interested in native plants? And at first, the best answer I could come up with was, well, you can find them on plants. You often find slime molds most frequently on decaying plant matter, whether it's leaf litter or decayed logs that have been broken down by fungi. Sometimes they're quite conspicuous. They can get quite large, up to the size of about a baseball, in some cases, for the ones you will find around here. So in that sense, they are relevant to plant people in the sense that you might find them when you're out for a hike, or botanizing, or working in your garden. Here's a photo of one of my favorite species, Diachea leucopodia, next to, I think, a male live oak catkin. Someone can correct me if I'm wrong. Yeah. So I got to thinking about the question of why are they relevant a little more, and I realized that you find them in your garden. This one on the right, the top right photo, Fuligo septica, also known as dog vomit slime mold, is quite conspicuous, and it pops up in gardens quite often. So here is to show how common it is. Of course, iNaturalist is down for maintenance right now. It's quite common. There's 101 observations in Pasadena alone, and it's seen quite often on mulch, wood chips, in landscaping, on grass, and it's large. It can be about the size of a baseball, and it's bright yellow, so it's quite striking. When people see it for the first time, they're like, "What the heck is that?" So in the slime mold identification and appreciation group on Facebook, occasionally there will be someone who's just heard about slime molds for the first time, and they come and ask, "What is this thing on my plants? Is it going to harm them?" And the answer is no. They're not pathogenic. They don't eat plants. They eat mostly bacteria. But it's also unknown if they're actually any benefit to plants. But no, they're not going to hurt your plants. There was an instance in the 1970s near Dallas, Texas, where people who had never seen these things found them in their yard and thought they might be an alien invasion. They kept trying to spray them with a hose, which of course just spread the spores. And these yellow blobs would pop up again in another part of the yard, only to appear bigger than they were the last time. So some people thought this is an alien invasion right here in Texas. There's these invincible beings that they just keep multiplying. They keep coming back as we kill them. So it gives you some sense of the surprise and even possibly hysteria people might display when they see these things for the first time right at their own home. But I think ultimately the best answer to the question of why should you care is because they're really beautiful and fascinating organisms. And I think knowing about them and tuning into them could enrich your experience with the wild areas you spend time in, or even your own yard. These are some of my favorite photos that I've taken, and some of the only ones that I've included in this presentation that are not taken in the San Gabriel Mountains, that I think display some of the most beautiful features of slime molds, and also just the diversity of their colors and their structures, their habitats, the organisms that are found on and nearby, and just how strange they are. So I've talked a little bit about the plasmodium, which is the slimy stage where slime molds get their name, the single-celled multinucleate creeping bacteria-eating mass that crawls around. And I've mentioned and shown the sporangia in the case of the garden slime molds, the Fuligoseptica, the dog vomit slime mold, which is a large mass of spores. And then there's also the orange sporangia that I showed, the fruit bodies that have stalks and little balls on the top that contain the spores. But I'll tell you a little bit about the rest of their life cycle. They start out as a spore, and that spore germinates, releasing 1 to 4 protoplasts, which are single-celled, single nuclei cells that either have a flagellum, which helps them move around, or they're more amoeboid and they change shape and move around. If the conditions are not favorable to them, they might turn into what's called as a microcyst. That cell goes dormant and waits for more favorable conditions until there's more water in the environment, there's more resources, and it's better able to move around. When they are active in the single cellular stage, they move around, and if they encounter another cell that's compatible, they will fuse. And at that time, the cell walls fuse and the nuclei also fuse. And when the nuclei fuse, a diploid zygote is formed. And this is your early plasmodium. So this early plasmodium crawls around, engulfs bacteria and digests them, metabolizes, grows, and as it grows, the nuclei synchronously divide. It still remains one cell, but there become many, many nuclei, and it becomes much bigger to the point where you can see it. Humans can see it with their own 2 eyes. It keeps crawling around, it keeps feeding. If conditions deteriorate again, if it dries out, if resources run out, it'll turn into what's called a sclerotium. The sclerotium is basically a hardened form of the plasmodium. It contracts, becomes more opaque, less slimy, contracts and goes dormant until it's better able to move around and keep feeding. Eventually, your plasmodium is going to be triggered by certain conditions such as sunlight, a change in pH, drying out of the environment, the local environment, increase in temperatures or freezing temperatures, or just lack of resources, and it's going to spoil it or form fruit bodies. This is the reproductive stage. So it forms maybe something, these stalked sporangia, fruit bodies with balls on them that contain the spores, or like the large yellow masses we saw earlier. And those fruit bodies mature and eventually release their spores, and the cycle starts again. So as I mentioned, depletion of resources or unfavorable conditions and also other less understood factors might drive this sporulation or formation of fruit bodies. And once it begins, it usually occurs over a period of about on the order of 24 hours to go from a plasmodium to a fully mature fruit body. Not always, it could take less time, it could take more time, but for reasons that I'll explain later, if you find a slime mold that's still maturing and you want to know what it is, it's a good idea to check back on it the next day, maybe 24 hours later, and just see how it's doing. And when I say environmental conditions drive this behavior, I'm talking about microclimates. I mean, we have had a very dry season for the most part here, but if you find the right spots with the right microclimates that hold moisture particularly well, you can still find slime molds. These are a couple photos that I've chosen because I think they really capture a few of these different stages of the life cycle. So here again, these fruit bodies, the bluish, almost spherical objects in the photo on the left, these are sporangia that contain the spores. And these are approximately 1.5 millimeters in diameter. You can see on one of the bottom ones, it's starting to crack open to release the spores. And all around them, this orange substance is the plasmodium that formed these fruit bodies. It underwent a irreversible chemical transformation to form these balls that contain the spores. Each individual segment of the plasmodium, each nucleus, essentially became a spore. But what's happening to this plasmodium is it's turning into a sclerotium. You can see that it's drying out. It's less slimy in some parts. It's looks harder. And this is the sclerotium, or the dormant phase, that protects the plasmodium, lets it wait out the conditions until it can feed again. In between the two larger orange sections, you see a segment of more active plasmodium, but this plasmodium is actually crawling into this crack in the log. And on the right, on the next day, you can see that the plasmodium has almost fully crawled into that crack to protect itself. So I first encountered this specimen 2 days before the first photo on a rainy day. It was just a plasmodium at that point. I came back 24 hours later. It had moved to a different part of the log and formed the bluish spheres you see. And then on the third day, in the photo on the left, the plasmodium had started to go dormant. And that day it had dried out, the rains had stopped, it was 10 degrees warmer, and it decided I've had enough, I'm gonna crawl back in my hole. So this kind of shows why it's interesting and beneficial to follow them over a period of multiple days. Not only this, but you can only fully identify them once the fruit body is fully mature. So as I mentioned, this process takes around 24 hours, just on average, kind of ballpark. And they can't be identified until the fruit bodies are fully mature. You need to see the characteristics of the fruit body. I took this time lapse last January on New Year's Eve of a slime mold species Bodhamia nitens, which I had never seen before, as the sporulation process was taking place. And in it, you'll see the plasmodium undulating, swelling in some parts as it forms more fruit bodies that look like the one in the center of the video. So you can see the blobs in the foreground are kind of growing and shrinking. Eventually, they may become more sporangia like the one in the center. And then in the background, you can see larger yellow sections that are farther along in the maturation process. But the one in the middle is still not quite there yet. It'll actually turn opaque and less shiny, more green and hardened once it's mature. But everything there is kind of on its way to the same stage. And there was a piece of material right next to this that actually had all mature fruit bodies. Yeah, the video was on a loop. I didn't get enough footage. This was taken over about 15-20 minutes, and I wanted to make it longer, so I did loop it, and it happens 3 or 4 times. So I was going to talk a little more about the anatomy of slime molds, but in the interest of time, I'll just mention the 4 different types. In the top left, we have sporangium, which we've seen by now. They usually have stalks with containers for the spores on top, and these threads inside, in most cases, known as the capolitium. The spores are attached to the capolitium originally, and the capolitium kind of gives the sporocarp, or the spore-containing structure, its shape, but it also helps push on the outer surface of the sporangium, the peridium, to break it open so that the spores can be released. On the top right, we have what's called an ethelium. This is dog vomit slime mold. It's a larger, usually larger, single mass of spores that has a single coating that can be broken open to expose the spores. On the bottom left is a not fully mature version of what's called a plasmodial, which is a fruit body that more or less maintains the shape of the plasmodium from which it came. So it stays kind of sessile or directly attached to the substrate and flat against the substrate with more of a plasmodial pattern. Then in the bottom right, we have a pseudoethelium. So it's actually a fused mass of individual spore containers that looks like a single mass, but they are individual. But I'll mostly focus on the sporangia and the ethelia in the top part of this slide. Like I mentioned, pretty much with a few exceptions, slime molds need to be fully matured fruit bodies to identify accurately. But in many, many cases, you also need to look at the microscopic features. So yes. Thanks, Margaret. Let me go full screen. I'm trying to move Zoom out of the way. There we go. So to identify many slime molds, you'll need to look at the microscopic features, such as the capolitium, these threads, and these spores. The spore ornamentation, these ridges you see in the spores on the left, are in the ridges you see on the threads, as well as those junctions in the threads. These are all important ID features that you need to look at to key out the species. So another time lapse to give a better idea of some of this anatomy and how it works. This is Arceria denudata, or cotton candy slime mold. I found it on a day in November where the Santa Ana winds had just begun. And I flipped over a log, and as soon as I flipped this log, the sporangia started opening, with the capolitium expanding and the spores becoming exposed, as it was exposed to the really dry air that day. So this one was also taken over a period of about 20 minutes. And you can see those capolitial threads, especially in the back, expanding very rapidly to break open that outer surface and expose the spores, but also expand more so that the wind can pass through there and carry the spores away. That one more time. So again, that was taken over a period of about 20 minutes, and it just shows how the environmental factors drive so many of these stages in the life cycle, and really just momentary changes are key to how these things live and feed and reproduce. So a little bit about the ecology of slime molds, or myxomycetes, which is a relatively unexplored field. This is mostly because they're infrequently observed in nature, except when they're in their spore producing or dispersing stage, when they're reproducing. In the microscopic stage, we can't see them in their natural environment. That's when they're swarm cells, or myxomycetes, before they fuse into a plasmodium. And even when the plasmodium becomes macroscopically visible, it's often hidden from the light and it's creeping around in places where we're not going to see it because it's eating. The dry air is going to be bad for it, so it stays hidden. And this means we don't really understand a lot of what drives the life cycle, since we only see it at this one part. And it's a beautiful part that we see it at, but it would be nice to know more about what gets us there and how to better seek out these organisms by knowing more about their. I'm going to go full screen again. More about their life cycle. So they can be found pretty much wherever there is decaying plant material and water. They like decayed logs that have been really broken down by fungi and leaf litter. They're predators, which is kind of surprising, given that we associate them with fungi. They crawl around eating bacteria and occasionally fungi too. We know that they move around, and in some cases they actually do help break down debris. They just by the fact that they contribute to that ecosystem, but not in the same way as fungi or detritivores. We also know that there is some substrate specificity. So some prefer logs, some prefer leaf litter, some have been only found on alder cones or alder catkins. But they're also not picky about what they fruit on. In the earlier photos of dog vomit slime mold in gardens, we saw that the slime mold actually left the mulch or left the wood chips where it was feeding, got onto concrete and said, "Hey, there's no food here. I'm gonna form a fruit by you. There's nothing here. I gotta go." So this photo on the right shows just how unpicky they are. This beautiful iridescent species, Laproderma scintillans, actually fruited on the head of a spider. It either died there or molted there. Now it was on an oleander leaf, I think, that I found in my neighborhood in Highland Park in Los Angeles. A few hours after a light rain. And it just struck me as kind of incredible about where they might pop up. Helena, I'm not sure how far they move. I know they've been known to actually crawl up trees quite a far distance, maybe several meters. But from my own observations, I've seen colonies of fruit bodies that are all fruiting at the same time over an area with maybe a 50, 60-foot radius on all kinds of surfaces. So it appears to me that, the same plasmodium may have been responsible for all of those. It may have covered that entire area, since they were fruiting over that entire area, or it might have split up after moving across that area into multiple plasmodia and fruited many times. So they really do cover quite large areas, but I'm not sure if anyone's tracked them over very great distances. To answer Kathy's question, I'm not sure how fast they move. you can kind of get an idea. I'm sure there's some information out there. I would say some plasmodium might move up to about 1 inch per hour. So they also have associations with insects. They not only serve as a food source for insects like this beetle on the left and this fly larva on the right, but insects are also serve as vectors for spore dispersal. So the insects will either eat the spores or rub on the spores. And as the insects either fly away or crawl away, they're able to disperse the spores over much greater distances than would have been possible through wind or water alone. So this beetle on the left, I see beetles of this family, I forget exactly what it is, with slime molds like this Arceria all over the West Coast, and it's apparently quite common. It was kind of just nestled into these sporangia as they were developing. And this fly larva on the right was part of a group of larvae that were eating this Physarum species. Now, this Physarum species actually forms an outer surface of calcium carbonate or lime, and it's like quite literally rock hard. It's almost difficult for me to pinch them with forceps or tweezers when I'm preparing a slide. So you can actually hear the crunch. So these larvae were kind of just tearing into them like scissors with their mandibles and eating the spores out of each one like a cereal bowl almost. And I felt as I watched them for a few minutes, like you can almost hear the larvae crunching on this rock hard slime mold. So presumably the larvae, they eat the spores, their stomach fills with spores. They eventually become a fly, and they fly away and carry these spores with them. Slime molds also have fungal associations. As I've mentioned, the plasmodium will eat fungi. So this yellow plasmodium in the top right was in the process of engulfing this crust fungus and presumably eating it. It may have been eating the bacteria on it, but some slime molds have eaten fungi. And you can tell after the plasmodium moves on from the fungus that the fungus has been digested. It's not in great shape. But slime molds are not the top of the food chain. Even though they are predators, they still fall prey to fungal pathogens. This fungus on the species in the bottom left is known as Polycephalomyces tomentosus, or Blistum tomentosum is a synonym. It infects members of the order Trichiales, and coats them with these white structures. And it's actually in the family currently classified with Ophiocordycipitaceae, which contains many insect pathogens and is somewhat related to cordyceps, which you've probably heard of. I'm not sure if the genetic research supports that classification, but that's currently where it sits right now. So it's interesting that it sits around a bunch of fungi that also infect insects. So now that we've had an introduction to slime molds, let's talk about how to go about finding them. The first thing I would start with is a loupe by hand lens or a magnifying glass, and or a clip-on macro lens for your phone, which you can buy pretty cheap on eBay. If you haven't seen them, it's just a little, I don't have mine here, but it clips on top of your phone camera and lets you get some nice close-up shots, which works pretty well with the phones we have nowadays. Or you can use a camera with a macro lens. I also use extension tubes. a magnifying front lens that gives me 2.5 times magnification, and many slime mold photographers, including a couple in the audience, well, my wife brought me one of the macro clip-on lenses. So you can buy these pretty cheap on eBay or Amazon. They just go right on top of your phone. They're good enough for getting some identification slots of slime molds. Thank you. Slime mold photographers use a method known as focus stacking to take a series of images at different focal lengths and then use software to combine those to get the nice crisp images that I've been sharing tonight. Where do you look? My favorite place to look is decaying logs. Look for logs that are well decayed but still intact, ideally in microclimates that hold a lot of moisture in or under the logs or in the area in general. I find that our canyons that face east-west, or like our transverse canyons, are pretty good at that around here. Millard Canyon, downstream from Millard Falls, is my favorite spot to go. It seems to hold a lot of moisture down there, and I moved to the area very close to there recently, and found an incredible diversity so far. But when you're looking at these logs, there's kind of a Goldilocks zone. You don't want one that's too dry, because there's going to be no activity, but you also don't want one that's too wet. It might even be too wet for the plasmodium. So a good place to look for these logs is riparian areas. I like to look for like partially submerged logs in the drier months, or logs where the stream or the creek has receded, and the logs are just sitting on soil where the creek used to be. I find that there's usually a good amount of moisture there that will give you a pretty good chance of finding something. Under dense oak canopies after a rain, if there's logs under there, that's sometimes helpful. I don't find a whole lot on coast live oak, usually alder and California bay laurel in our area. But you can also find them on leaf litter after the rain. So to answer Elba's question, which was just posted in the chat, yes, you can find them anywhere. Some of my very favorite finds have been in Los Angeles, in urban Los Angeles, after a rain. Native plants, yeah, you'll find them there, but also ornamentals, anything in non-native landscaping. Some of my favorites are English ivy, oleander, because they both hold really well sequester amounts of moisture in their foliage at the bottom, and Chinese elm. For whatever reason, Chinese elm leaves have shown me some really interesting species, like the iridescent Lamproderma species. Starting this time of year, February through about April, you can see my favorite species, the bright blue iridescent Diachea leucopodia, on alder leaves. I see a lot of this at Millard Canyon up above the falls. Here is a photo of me at a log at Mount Shasta last year. This log is kind of on the upper end of how decayed you want. You can see some parts of it are pretty broken up. So you want, there was a lot on here, a lot of really interesting fungi and slime molds, but look for something with this level of deterioration kind of as your upper limit. And this video here, I took almost directly under the Colorado Street Bridge at Lower Arroyo Park. And it kind of shows some of the substrates that you will find slime molds on. This was in a patch of eucalyptus debris, and there were 5 or 6 species that I found within 15 minutes on bark, on wood, on leaves, on twigs, kind of just all over this maybe 50-foot area. So you can see on this piece alone, there's multiple species. One that my camera's pointed at, and then this other one, which is the yellow species I showed a time lapse of earlier. There's a plasmodial carp on that twig. I'll play that one more time. I think this really gives you a good idea of kind of just a basic idea of what you're looking for. I mean, that piece of bark is also on top of a log that has 2 more species on it right there. So that's right under the Colorado Street Bridge. It's a great spot to look after a rain. And lastly, where to find slime molds? Near other slime molds. At least about half of the time when I find one slime mold species on a log, I will see at least one more slime mold species fruiting at the same time on that same log. So here in the photo we have Craberia violacea. It's a very, very tiny species. And in the background, the slime mold behind it would actually tower over it if it were closer, is a Trichia species. And they were fruiting at the same time. And this is yet another time lapse that shows a plasmodium engulfing a few sporangia of a Comatricha species. So the plasmodium is still kind of undulating and throbbing as it feeds, digests, grows. It's not moving right now a whole lot, but the plasmodium is very much still active, and it's a different species. I can tell that much by the pigmentation and the density of it than those sporangia that it's engulfing. I'm not sure if it's eating those or something that's around them, but it's not out of the question that it's eating them. So if you see slime molds fruiting, take a very close look, keep looking, because you're likely to find more. So a little bit about what we have here, since this is a San Gabriel Mountains group. Here's a map I pulled from iNaturalist showing the distribution of Myxomycete observations, kind of centered on our mountains. Obviously, there's more observations down in the foothills because people are observing there more, but you can find the slime molds all the way up to 7,000 plus foot. Pretty much anywhere. I mean, even up getting towards the top of Mount San Antonio. What I find is remarkable is that I've found and identified confidently over 50 species in the San Gabriel Mountains in about the last 2 years since I've been looking. And what's remarkable about that is that there's only about 1,000 species worldwide. So that's 5% of all slime mold species that I've seen one person here so far, and I'm sure there's more. And those 50 species that I've seen represented 25 genera of the 52 that are worldwide. I think this answers one of the questions we had in the chat. Are they widespread? Can you find them in the city? Yes, they're widespread. You can find them pretty much anywhere. Many species appear to be cosmopolitan, meaning you can find the same species all over the world, at least the same species in many temperate climates, or the same species in many tropical climate habitats. So it appears that a lot of them may be like this. You can find them anywhere, but they're loosely separated into temperate and tropical climates. This isn't a hard rule, but there are a lot of species that are all over the place. Some rare European species have started to pop up here in North America as slime molds are starting to get more attention, thanks to social media. Some pretty interesting headlines about fascinating research that's being done with them. Some very sci-fi sounding headlines, and people who are trying to get the word out about them. So there have been some pretty exciting jumps in the range of some slime mold species in recent years. So I want to show you a few of the most common species that I see here in the San Gabriel Mountains. This is Reticularia lycoperdon. It's named after the fungal genus Lycoperdon, which is puffball mushrooms. So this slime mold is colloquially known as the false puffball. It's quite large. It's around the size of a quarter to this one that was nearing the size of a baseball. And you can find it on dead alder logs that are not as well decayed as some of the others I've shown. Sometimes live alder, sometimes live oak logs. And it's quite large and quite common. Another very common one is wolf's milk slime mold, Lycogala epidendrum. When it's still maturing, it forms these pink balls on the side of logs that are about a centimeter, maybe a little larger or smaller in diameter. And when I find them, they're really fun to pop open. So here is me popping a slime mold pimple in slow-mo. This one's not quite mature yet, so it's still slimy on the inside. If you ever come across these, they're pretty common. I highly recommend popping this. It's very satisfying. And it's just a fruit. It's not the actual organism or anything that's left, since these plasmodia, when they form fruit bodies, it's an irreversible change, so you're not really harming the organism except for preventing that single one from dispersing its spores once it matures. Here is a couple species of Stemonitis. It's another relatively large slime mold, and the fruit body is relatively large. It is often bright pink on the side of logs. It has these almost hairy black strand-like stalks with kind of tubular white or pink sporocarps on each end. Sometimes they fuse together. So on the left, this one's not quite mature yet, but on the right are two photos of a mature specimen. Before and after I blew the spores out. So in the bottom image, you can see the network of capillary threads where the spores were attached. I want to answer this question before I forget, because toxicity and allergic reactions are important. Yes, you can have allergic reactions to them. I've never heard of this happening, but as with anything, people can have an allergic reaction to this. As far as I know, none are toxic, and a couple of them are actually eaten. So if we go back to Helena asked me this question earlier as well. If they smell, I've never noticed a smell from one, but Aaron, they're in the audience, they can vouch for this. I tried to eat this one here. and it was disgusting. It looked like chocolate on the inside, and it was horrible. But apparently, when it's less mature, before the spores are fully formed, it is eaten in Veracruz, Mexico, and it's called caca de luna, or moon poop. It's fried up like scrambled eggs. So, as far as I know, none of them are toxic, and some of them are eaten. Dog vomit slime mold can be eaten as well. If you want to try it, go ahead. Probably when it's still in its bright yellow stage. And I know the name dog vomit slime mold doesn't really make that sound like a great idea. And neither does moon poop. Here we have Steminatopsis typhina. This is a very common slime mold here in the San Gabriel Mountains. It's slightly taller than others, maybe 3 or 4 millimeters tall at times. It has this almost like hot dog on a stick look to it, especially when the spore carps get a little pinker than they are in the right-hand photo, where you have a small little snail for scale. The snail was about, oh, thank you, Leah. Dog vomit slime mold should not be eaten because it can concentrate high amounts of zinc, which is not good for you. I'm not sure about the effects, but Leah's recommendation is don't eat it. And she's more of an expert than I am, so trust her. Comatricha nigra. This is a also common species. In this, I find it year-round if the conditions are right. Even in the middle of July, if we get a day where there's a good fog layer or a good amount of moisture in the air in the mountains, you'll see it fruiting on logs in the middle of summer. This top right photo is a great example of the gradient of color and shape that the species has as the sporangia develop. I flipped over the logs so it's upside down, but the darker ones on the bottom of the top right image were more exposed to light, so they're farther along in maturation. And the ones on the top are lighter colored. They were originally in more shade. As they mature, they change from white and elongated down their stalk to a pink salmon color, to a bright pink, to a brown, to a black, and then eventually the black outer layer dissolves and the chocolate brown spores are exposed. You can kind of see a brown one right here with the spores just barely exposed. I'm pointing out a mistake in my photo because it was a photo stacking or focus stacking error, but there is one right there. Here we have Arceria denudata, or cotton candy slime mold, which I showed in a previous time lapse. In the bottom right, you see it immature. It's white, kind of in the crevice in the log, still developing. Arceria ovulata, a very common slime mold that holds up quite well. So if you pull a log apart any time of year, you might find this. It'll kind of spring down as you take a piece of bark off and dangle there. The orange background on the left is a fungus known as, I think the current name is Ceroporia spissa. It's a bright orange crust fungus you may have seen before. And here, my absolute favorite slime mold species that you can find here is Diakia leucopodia. This is just a truly beautiful species. I think Leah's mentioned that it's her favorite species as well in the past. It's common right now if we get enough moisture in the canyons. I mentioned before, Millard Canyon's a great spot to look for it. and you'll start out seeing it as these white stalked, white sporocarped fruit bodies that eventually develop this blue iridescent outer layer. And from the top down, you can see this white network connecting each sporangine. So that's something to look for if you're looking for it. If you're looking at it from our level. So this one is not actually a Myxomycete, it's in a sister taxon. The name's escaping me right now. But this is Ceratiomyxa fruticulosa, coral slime mold. And it actually, instead of containing spores within each of those strands, it puts a single spore out on a single stalk. Each of these strands has many stalks. Each has a spore at the end of it, outside of the structure. Thanks, Leah. Protosteliomycetes. It is usually lumped in with slime molds, and I include it here because it's a nice one to look for. It's a great indicator of other slime mold species in the area. If you find this on a log, just keep looking because you're in the right spot. And this can form some pretty extensive fruitings. So wrapping up a little bit, as I've been exploring these organisms in the San Gabriel Mountains, there's a few questions that have come up in my head that I want to explore more. First, tying it back to native plants, do they form any associations with native plants? Are there particular slime mold species that seem to prefer particular native plant species as their substrate of choice? On the right, I have a photo of a species that I'm going to try to grow in a moisture chamber that I've encountered on live oak a few times, coast live oak logs, which is a substrate that I rarely find any other slime molds on. So this one stuck out to me. And it just has a really interesting morphology, and I'm not sure what it is. I'm trying to track it down, and it really seems to prefer our most ubiquitous and kind of famous native plant, Quercus agrifolia. So what other slime molds are making these kinds of associations with native plants? And what does that mean? Next, there are a lot of slime molds that fruit and live their lives in snowmelt. So in the spring in the mountains, when the snow starts to melt and the temperatures start to rise and the forest floor dries out, slime molds will start to fruit. Leah, if you look here and next to it, there's a little bit of plasmodium that seems to be associated with it. I have found it in sclerotium stage as well. Inside, like deep inside of oak logs. But continuing with the snowmelt nipicola species, they fruit when the snow melts, and there's a lot of really interesting slime molds that can be found in the spring. So my goal for the next 3 months, essentially 4 months, hopefully, is to document as many of these I can, especially here at home. When I visited Oregon last spring, I went to a fire scar and found a particular Comatricha species on almost every dead, burned conifer, especially Douglas fir, that I saw. Margaret Gallagher and I went looking for some of these last year in the bobcat fire, and we didn't see any. We got a little distracted by some other organisms we found. But I would like to find out if this Comatricha species also occurs here in the bobcat fire. Then also, what about desert and chaparral slime molds? There is at least one Bonhamia species that fruits on a puntia species of cactus, prickly pears. There's other desert slime mold species, but they're a little hard to find because you got to time it right and be in the desert when it rains and be in the right spot, and not much is known about these. So I'm thinking on the north side of the mountains, or possibly out by Joshua Tree this spring. I would like to see what's out there, but also what's in our local chaparral habitats. I haven't found slime molds in chaparral yet, but I'm sure they're there. So just a little more on this. I'm hoping that with the information you've been given here tonight, and hopefully a little bit of interest in these things, you can go out and document them, post them on iNaturalist. You can send them to me on Instagram or tag me on iNaturalist. I have the same handle on iNaturalist. I'll look for them either way and try to identify them. But it would be great to get a better idea of just how much diversity we have here. I've already seen that we have 5% of global species here in the San Gabriel Mountains, and I'm sure that's not even close to the end of it. So this is kind of a call to action. When you're out looking at native plants, take a look, keep an eye out, you won't regret it. So some resources in case you're interested in going a little deeper. On social media, Alison Pollock, I think I saw her in the audience here tonight. She's an incredibly talented macro photographer who takes the most beautiful pictures of fungi and slime molds. She's in Marin County. Her handle is Marin Mushrooms. She posts, she's kind of helped build the hashtag Slime Mold Sunday, which has gotten a lot of people into slime molds. She's been a great mentor to me with my photography, and I highly recommend taking a look at some of her photos. Erin just posted Allison's Instagram in the chat. Sarah Lloyd in Australia is kind of the OG slime mold influencer on social media. She's also not just an influencer, she's a scientist. She's written 2 books. One of them is on the right-hand side of the slide here. And she has also been a great guide to me as I've gotten into these organisms. She's a wealth of knowledge, and her books are beautiful, her photos are beautiful. She's doing making a lot of really important ecological observations that have not really been made before, and asking questions that no one else has yet. Leah Benlin, who's also here tonight, she's another great fungi and slime mold educator who also does talks like this. I watched a talk of hers a few weeks ago where I drew some inspiration for this talk from. And she posts some really, really interesting fungi and slime molds on her Instagram as well. And she started the Slime Mold Appreciation and Identification group on Facebook, which is a great resource for posting photos of slime molds for experts to identify, or just to get information. I mean, browse the posts and look at photos, or read some of Leah's very informative posts about the slime mold life cycle and really deep down questions. And then lastly, there's Barry Webb in the UK. He's another incredibly talented photographer who posts a lot of really beautiful close-up photos. And feel free to contact me on Instagram or my email. I'd be happy to answer any questions about where to find slime molds, what your slime mold finds are, or just any questions you think of after this talk. There's a few books I wanted to shout out that I've used, some of which I've used for references here. Myxomycetes, A Handbook of Slime Molds by Stephen Stevenson and Henry Stempin, and Where the Slime Mold Creeps by Sarah Lloyd. These I've both referenced both of them heavily in this talk. They're great introductions to the biology of slime molds. They have introductory keys to start identifying slime molds. If you want a more comprehensive book, a guide to temperate myxomycetes by Nananga Brahmakamp is a great resource. It has more comprehensive keys and better information for identification of more species. Allison mentioned that Myxomycetes, a handbook, has just gone out of print, but there are still some available at booksellers. I saw several copies at Powell's Books in Portland when I was there a few weeks ago, so you can order from them online. And then this is a French name, I'm going to butcher it, but Les Myxomycetes by Poulain, Marianne Meyer, and Jean Bosonnet. It's also out of print, but it's a extremely comprehensive 2-volume set, one of which is all photos, and the other of which is all keys of almost every slime mold species that's been described so far. It was worth my time and money to find these and buy these. They're an invaluable resource if you really want to go deeper into Myxomycetes. And lastly, my friend Erin is organizing an event at Arlington Garden Lot next Friday night at 7 p.m. where they're going to be doing a showing of a documentary called The Creeping Garden. I have not seen this documentary. I hadn't heard about it before, but the trailer looks fascinating. And it looks like it's going to cover some of the more interesting and inventive slime mold research topics that people have been exploring in recent years. And Erin just posted a link to the Eventbrite for next week's event at Arlington Garden. Thank you. And that's all I have. Thank you. I think I went a little bit over time, but I'm hoping you all enjoyed it. I can take some questions now. Margaret, do I think there are many undescribed species? Yes, I do. And speciation is also weird. I suspect that there's a chance that the species here that I saw on Coast Live Oak, there's a chance that it is undescribed. I haven't been able to find anything like it in the research, in the literature. But yes, I think there are probably many undescribed species, but it's also a fairly small group of organisms. I mean, it's only 1,000 known species so far. I don't think that's going to get too much bigger. They've been relatively stable. The oldest slime mold fossil that's been found so far is about 100 million years old. And it may even be the same species. It was a Stematitis species as ones we still have. So there's not a lot of speciation occurring, possibly. So I wouldn't be surprised if the number of species doesn't grow too much. Hannah and Tyler asked, have you found a slime mold you couldn't identify? All the time. I have a whole stack of slime molds that I am meaning to get under the microscope and identify, but I probably won't get to all of them. Some of them get really tricky to identify. Is there any place in the world that I would like to travel to look for slime molds? I want to go to the tropics soon. I've never been to the tropics, and I would like to see what's there. I'm sure it's pretty easy to find them there. How do they actually eat bacteria, and do they form some kind of vesicle? I'm not sure how to answer these questions. They don't form any kind of vesicle. as far as I know, I'm not sure about the digestion process. Enzymatically, they are single-celled organisms, but they do engulf bacteria. So they bring them inside the cell. How do you collect and store them? Preferably you find a sclerotia or a plasmodium, and you can bring that home and put it in a moisture chamber and try to get it to fruit and form fruit bodies you can identify. I collect them and store them in, this is a jewelry case, but that's the same thing. Specimen boxes like that. You can send them in matchbooks. If you want to send them in the mail, you can send them. Oh, I'm trying not to share my screen. Oh, my wife is giving me tips here. I have logs right next to me that I've collected that have fruit bodies on them, sclerotia still in them. Then I'm gonna try to get to fruit at some point. It's kind of all over my office. How do they consume food? I think I kind of covered that. What about large geographic distributions of the ones in your photo? How widespread in the state and country? A lot of the ones that I shared tonight, I've seen all over the West Coast. And they probably can be found much wider than that as well. Are there any other questions I'm missing? Do they produce waste? That's a great question. I don't know the answer to that. Some species have a slime sheath as part of their plasmodium. It actually leaves little, sometimes black or red tracks on a log. If you flip over a log or take bark off a tree, you can see black tracks where the plasmodium was. I suppose this is waste in a way, or it could be waste. Oh, my camera and lens setup. I use an Olympus OMD series camera with a 60mm macro lens and a very cheap tripod. I've just started using flash. I use a magnifying front lens on the front of my lens, extension tubes to get closer, and I use the internal focus bracketing that the Olympus cameras have. So it'll automatically take up to 1,000 photos in focus sequence that you can then stack in another piece of software. Another question I missed was, are there invasive slime molds? I don't think I can answer that question. I mean, it's hard to say what's invasive, and we don't know enough about the distribution of them because they've been so infrequently observed to say that one's not native somewhere. Or how it got, if it's not native, how it got from one place to another, like the ones we've been seeing that are rare in Europe pop up in North America. Are they invasive? I mean, to my knowledge, they're not detrimental. And I think something would have to be detrimental to native ecosystems to be considered invasive. So I'm gonna say no. And I think that's all the questions I've seen so far. Thank you again. It was a pleasure getting to share something that I'm obviously very excited about, and I hope you are too now. Thanks for inviting me, even though it's not plants.
Well, thank you, Stu. It was really great. Love it. I'm fond of slime molds too. So thank you all for coming. Our next meeting will be on March 24th. Same time, same place. So see you there. Now I have to go out and look for slime molds again. Bye.